During 2018 summer, we found a large number of empty snail shells scattered on the ground during an investigation to identify biodiversity underneath rotten wood. After close observation, we discovered that these snails were killed by some flatworms crawling within centimeters beside them. However, we couldn't identify the species of these flatworms under any planarian catalog in Hong Kong, so we started comparing them to catalogs from other regions and found that they closely resembled New Guinea flatworms. In awe of the discovery of this new species of flatworms that we've never previously encountered, we wanted to concretize their identity. Therefore, we identified them using the traditional morphological classification method. We looked at the flatworm's physical attributes. They are flat with both ends pointed and are 60 to 75 millimeters long, 3 to 5 millimeters wide, and 1 to 2 millimeters thick. The dorsal side of the flatworm is dark brown with a yellow slender central line, and its ventral side is pale gray. These features match exactly to the morphological classification standard of New Guinea flatworm. As we've identified the species we found, we started conducting some online research on the species of flatworms. We found that it is extremely invasive and has caused the extinction of countless native land snails on the Pacific Islands. Therefore, it is the only flatworm listed among the top 100 world's worst invasive alien species. According to the latest distribution diagram of New Guinea flatworms, they have appeared in more than 15 territories globally, but are not distributed in Hong Kong, even China. Thus, we are certain that this is the first report of New Guinea flatworm in Hong Kong as well as China. To understand their predation behavior, a comparative experiment was carried out to test the flatworm's food preference among several invertebrates. Our results show that New Guinea flatworms have a selective prey preference on local land snails, especially the native tree-inhabiting snail, rather than other species like the invasive giant African snail. Therefore, the existence of New Guinea flatworms may not only cause the local snail species to decrease and even slowly go extinct, but also assist invasive snail species in expanding their colonies, leading to an imbalanced ecosystem and potential economic losses. After recognizing their harmful behaviors, we decided to explore the severity of their invasion in Hong Kong in order to analyze their potential impact on Hong Kong's local ecosystems. The distribution of this flatworm was therefore investigated in multiple locations, as shown by the colored regions. We found that New Guinea flatworms have already invaded central and the southern districts of Hong Kong Island, shown by the red regions, but not any other regions. We then randomly chose 3 1-square-meter areas as quadrats to measure the flatworm's distribution density in the occurrence areas. Based on the sampled results, the total number of New Guinea flatworms was roughly estimated to be as many as 17,100 within the investigation area of 3,240 square meters in the Southern District area, which is 5 flatworms per square meter. We found that New Guinea flatworms have been mated with sparse distribution but a high density in the southern region of Hong Kong Island. They may be a severe threat to local snails and even the entire ecosystem if we do not take any effective approaches to control them. We wanted to present our findings to more established experts of the field and demonstrate the significance of flatworms' invasion to them. Disappointingly, our results were not acknowledged due to the lack of recognition and morphological evidence. Yet, having the strong urge to prove our findings, we contacted Professor Hu and received the opportunity to conduct molecular analysis inside a laboratory as a more concrete measure of identification. We first captured the New Guinea flatworms and fixated them in alcohol. We then extracted their COX-1 gene by digesting part of its body using enzymes. And filtered the DNA through centrifugation. After filtration, we cropped out the specific COX-1 DNA using 2 primers. The COX-1 gene is chosen to be sequenced because it is part of the intermembrane protein on the inner mitochondrial membrane as a part of the electron transport chain of photosynthesis. Every species has a unique mutation of the gene synthesizing this protein, and the COX-1 stays almost identical within the same species for a few generations. Therefore, we can identify different species by the COX-1 genes. We then amplified and cloned the set of COX-1 gene we extracted and ran it through an electrophoresis gel plate for gene purification, leaving the COX-1 gene to be sequenced. By comparing the COX-1 sequence we had and the submitted sequence of New Guinea flatworm from the GenBank, we got an identical match with the New Guinea flatworms labeled as world haplotype, while getting a 95.02 to 96.27% matching with the New Guinea flatworms labeled as Australian haplotype. While we were collecting New Guinea flatworms for molecular analysis, we coincidentally found another species of point-headed flatworm that looks quite similar to New Guinea flatworms. Comparing its morphological features to that of the after looking at various animal catalogs from different regions, we couldn't pinpoint their identity either. They're very similar to that of New Guinea flatworms, except that they have 2 yellow stripes along their dorsal side rather than 1, and the tip of its head comes in transparent orange color. Are they a new type of species? With questions in mind, we conducted molecular analysis and found that the 2 species only have a COX-1 genetic similarity of 86%, and the new species having a similarity of lower than 95% with all other species recorded in the GenBank. Thus, we came to the conclusion that this species is indeed one that has never been discovered before. We report here, for the first time, the occurrence of New Guinea flatworms and a new species in Hong Kong. The invasive species New Guinea flatworm certainly brought serious threats to the local ecosystem and economy. Considering this, it is time to take action to monitor its negative impact and control its spreading to regions nearby. These findings provide updated information on the global distribution of New Guinea flatworms and its detrimental effects on local biodiversity, as well as raising public awareness to allocate more resources to conducting scientific research on the efficient methods to control this species. At the same time, the biodiversity regarding flatworms in Hong Kong is updated with the discovery of this new species, helping animal biologists achieve a more comprehensive view on the variety of land planarians in Hong Kong.